Apoptosis means we know that it is a programmed cell death. In shortly, cell suicide. And the other two things that we have to remember in apoptosis means there will be no inflammation and no leakage. Mechanism of apoptosis. There are two pathways, the extrinsic pathway and intrinsic pathway. These are further divided with the initiation phase and execution phase. Intrinsic pathway is also divided the initiation phase and execution phase. First, we'll discuss about extrinsic pathway. What will happen in the extrinsic pathway of initiation phase means this is cell, this is the nucleus, and this is the DNA. The signals come from outside of the cell to commit the cell suicide. These are called as fast ligand. There, there are the receptors, which are called as the death receptors, present all over the cell membrane. These external signals called as fast ligand bind with the receptors, which are called as death receptors. Fast receptors are the CD95 and the TNF receptors present on the cell. They form the domains. All the receptors, when the fast ligand or the external signals come and bind to the fast receptors, they will form the ligand called as fast associated death domain. And when the TNF signals or the TNF ligand comes and bind to the TNF receptors, it will form TNF receptor associated death domain. These will activate the procaspase 8, and then the procaspase 8 will activate the caspase 8. These are the enzymes present in the cytoplasm, which will lead to the apoptosis. And now the intrinsic pathway. Initiation phase. As in the extrinsic pathway, the signals are coming from the outside so that the cell can commit the suicide. In the intrinsic pathway, the signals come from inside of the cell, from the damaged part. When the DNA get damaged, it will send signals to the mitochondria so that the cytochrome C is released into the cytoplasm and initiate the apoptosis, where actually the cytochrome C is present in the intermembranous space. There are mitochondrial pores present in the outer membrane of the mitochondria for the leakage of the cytochrome C into the cytoplasm. Normally, the cytochrome C remains in the intermembranous space by the anti-apoptotic molecules called as BCL2, MCL1, and BCLX. These are the anti-apoptotic molecules which are present so that they will not release the cytochrome C into the cytoplasm, so that the apoptosis will not initiate. And when the DNA get damaged, it sends signals to the mitochondria to release the cytochrome C into the cytoplasm. So these anti-apoptotic molecules are replaced by the pro-apoptotic molecules by the mitochondria. What will happen means the replacement of the anti-apoptotic molecules. Then activation of pro-apoptotic molecules like BARC, PARX, BIM, and BAD. These are the pro-apoptotic molecules which are activated. Cytochrome C is released from the mitochondria, and then it binds with the APF1 molecule which is present in the cytoplasm and activates the procaspase 9, which will activate caspase 9. This is the enzyme which is activated from the intrinsic pathway, and from the extrinsic pathway caspase 8 is activated, which leads to the apoptosis. What happens in the execution phase means, which is same for both extrinsic and the intrinsic pathway. From the intrinsic pathway caspase 9, and from extrinsic pathway caspase 8, they together will activate other caspases in the cytoplasm like caspase 3, 6, 7 in the cytoplasm, and activates all the other caspases in the cytoplasm and forms the apoptotic bodies, which contains a cytoplasm and the membrane and also some part of the nucleus. These apoptotic bodies undergo phagocytosis and ultimately cell death.